Have you ever wondered how you grow? Organisms are made of cells, the building blocks of life. In order to grow, you need more cells. How do cells form? They use mitosis and divide, one cell turning into two cells turning into four, and so on. By the end of this lesson, you will have a basic understanding of mitosis and be able to identify the different stages of cell division. Before any dividing happens, a cell is in a resting stage. In this stage, called interphase, the cell grows. It prepares for mitosis by gathering nutrients and energy, as well as one other important ingredient, DNA. DNA is hereditary material that lies inside of the nucleus of a cell. In order for a cell to divide, it has to have all of its instructions, or DNA, copy itself so that both new cells have all the genetic material. When you look at a cell in this phase, its nucleus is almost completely black. Now, mitosis is ready to begin because the DNA has duplicated itself. In the first stage of mitosis, called prophase, DNA thickens, coils, and pairs itself with its duplicate to become chromosomes. They become short, stubby rods in X shapes. The nuclear membrane disintegrates. You can easily recognize this phase because it looks like there are worms in the nucleus of the cell. In the second phase of cell division, the chromosome pairs, connected by the centromere, line up at the equator of the cell. Spindle threads form and connect the chromosomes. This stage is called metaphase. A good way to remember this stage is because metaphase starts with an M, and so does middle, because the chromosomes move to the middle of the cell. This phase is easily recognizable because it looks like the cell has stitches down the middle. Anaphase, the third phase of mitosis, begins with the spindles lengthening and making the cell longer. The threads pull the chromosomes toward the opposite ends. Half of the chromosomes go to one side, and the other half go to the other side of the cell. This is why the DNA duplicated, so that both of the new cells will have a complete set of DNA. When a cell is in this phase, it has lines on both sides of the cell. During the fourth phase of mitosis, the telophase or telophase, the chromosomes regroup and they begin to organize themselves into two nuclei. The nuclear membrane forms around them. A dent or furrow begins to form down the center of the cell. You can identify this phase by observing the two bunches of chromosomes, the nuclear membrane and the slight dent in the middle of the cell. The fifth and final phase of mitosis is cytokinesis, and it completes the cell cycle. For animal cells, the cell membrane separates the two nuclei in the cell to form two new cells that both have a complete set of DNA. For plant cells, the cell plate separates the two nuclei in the cell to form two new cells that both have a complete set of DNA. This phase is recognizable because you can see the cell membrane or plate forming between the two nuclei. Can you guess what comes after the last stage of cytokinesis? The process starts over, beginning with one of the cells in the interphase stage. What stage of mitosis is this cell in? That cell was in telophase because you can easily see the two nuclei, but the cytoplasm has not split yet. Do you know what stage of mitosis this cell is in? If you said prophase, then you are correct. You can easily see the chromosomes, which look like worms, in the nucleus of the cell. What about this cell? Can you identify the stage of mitosis this cell is in? This cell is in anaphase because you can see the chromosomes are moving away from each other. I hope you learned a lot and enjoyed watching this video. Thanks for watching and have a nice day.